Hello everybody, we are Group 4 and today we're going to talk about a rice breeding breakthrough. My name is Lino Calderero.
My name is Carson Orpin.
My name is Riley Carr.
And my name is Luke Labar.
One staple food crop. Currently, the majority of rice is grown in developing countries. Climate change and its effects threaten the security of this crop for the world's poor. The hybrid varieties that provide more vigor, strength, and therefore higher yield are unavailable to poor farmers because they are too costly. The article we found, Rice Breeding Breakthrough Could Feed Billions, by Andy Fell, discusses how the advantages of a hybrid rice genome can be gained without the high cost by cloning hybrid genomes from generation to generation. The article highlights the advantages of a hybrid genome to avoid inbreeding effects, how modification of mitosis and meiosis are the key to cloning hybrid rice, and finally, how CRISPR-Cas9 was used to make these important edits in the rice genome.
So right now I'm going to show you the downside of hybrid crossing. So the parent generation, one of them is homozygous recessive for productivity, but it is homozygous dominant for disease resistant. And the other parent is homozygous dominant for productivity, but homozygous recessive for disease pressure. So whenever you cross those two, the F1 generation is heterozygous for both productivity and disease resistance. So the offspring of that are going to show great productivity as well as disease resistance. So this Punnett square is showing crossing of two F1 hybrids. So what I'm doing now is going through and circling all of the offspring from the F1 hybrid cross or the F2 progeny that have great productivity and great disease resistance. In this case, the green skittle represents great productivity or yield, and a red skittle represents great disease resistance. As you can see, 9 out of the 16 offspring have both great productivity and great disease resistance. This is only 56% of the progeny that are showing the traits that were breeded into the F1 cross.
Meiosis is a process similar to that of mitosis, as the first steps are similar, except in meiosis, crossing over occurs during late prophase 1. After anaphase 1, enterokinesis occurs, followed by metaphase 2, where the individual chromosomes align. Then, at anaphase 2, chromosomes separate, creating 4 haploid genetically variable cells. Mitosis is the process of nuclear division beginning with interphase, where the cell duplicates its chromosomes in preparation for division. Then in prophase, chromosomes condense where each chromosome possesses 2 chromatids. Next, in prometaphase, the nuclear membrane disintegrates and the spindle microtubules attach to the chromatids. Then, metaphase is where the chromosomes line up on the metaphase plate and the spindles are still attached. The sister chromatids then separate and move towards opposite poles during anaphase. Finally, in telophase, the chromosomes arrive at spindle poles in the nuclear membrane, creating 2 identical cells.
So what scientists have done to induce mitosis in these cells is, first what they do is they find the DNA sequences that differentiate meiosis from mitosis. And once they get those sequences figured out, they make up a tDNA that acts as a guide DNA, and they insert that into the egg embryo along with the Cas9 protein. And they insert that into the egg embryo of the rice, and that goes and floats along until it finds its target DNA sequence. And the guide DNA tells the Cas9 protein where to cut, and when the Cas9 protein cuts that, and then the Cas9 protein keeps moving along until it finds those 2 other mutations to cut. And what scientists have also done is, through homologous direct repair, is a new DNA sequence is created and is inserted into the DNA sequences. When that's inserted, the new DNA sequence can then be recombined, and the product is we have a new DNA sequence that acts as mitosis by producing genetically identical offspring or clones through asexual reproduction or apomaxis.
Scientists found that 95% of the edited rice plants were apomixatic after 3 generations. This elegant gene editing process allowed 3 modifications to be made to the rice genome, causing it to reproduce asexually, cloning its hybrid genes, and passing the hybrid vigor and the reversal of inbreeding effects down generation to generation to generation.
Okay, the hot dog.